My pleasure to introduce Dr. Shelbi Russell. So Shelbi did her BS, her Bachelor's of Science, at University of Kansas at Lawrence. Is that the only Kansas?
Just one location.
And then went on to do her PhD in the Department of Organismic and Evolutionary Biology at Harvard University. I had the great pleasure of being a graduate student beside her. And then she has now moved on to do her postdoc at UC Santa Cruz in the Department of Molecular, Cell, and Developmental Biology. You're starting your second year there?
Yeah.
And now she's actually the UC Chancellor's postdoctoral fellow in the lab of Dr. Sullivan, Dr. William Sullivan. And the thing I really appreciate about Shelbi's work is that I am of the frame of mind where I really think like in the modern era of evolutionary biology, we're going to make the fastest strides, the largest strides at the intersection of disciplines that have typically historically operated largely on their own. And I think Shelbi's work really sort of highlights the sort of things that can be interfaces. So her work, really, I think, traverses the boundary between, evolution and ecology, development, and all punctuated by, I think, some rather, all towards understanding sort of how the mechanisms and processes of eco-evolutionary dynamics as they play out in microbial systems. So I'm really excited to hear about her most recent work. So please join me in welcoming Dr. Shelbi Russell.
Thank you so much, Shane. Thank you all for being here. Thanks for having me out. I'm excited to share my work with you. This is work I've done over the course of my PhD and postdoctoral career to understand how symbionts are transmitted between host generations, which often involves the germline, and as I allude to here, it can be a rather complicated, arduous process, such as hitchhiking through the galaxy might be. So I'm going to start out with defining symbiosis, and I really like the definition coined by Anton de Berry in 1879, which is the living together of unlike organisms. And the reason I really like this definition is because I feel that it captures the diversity of associations very well. So we see symbiotic associations all over the Earth, from terrestrial environments, such as I've shown here, with plants that associate with root mycorrhizae, fungi, that help them obtain water and nutrients from the soil, to the bottom of the ocean, along the sea floor at spreading centers, where you see these hydrothermal vent communities with huge amounts of biodiversity and biomass that have been enabled to live there through association with bacteria and invertebrate animals, to the tropics, where we see an enormous diversity of parasitisms between animals and protozoans in animals. And as these examples illustrate, I really like thinking about symbiosis in its most basic form, and that includes associations that span this cost-benefit spectrum. So you can have associations where one member benefits at the cost of the other. These are termed parasitisms or pathogenic interactions. And then at the other end of the spectrum, there are mutualisms where both members benefit from the interaction. Then in between these are commensalisms, where they're living together intimately, but there's no net gain or loss due to the association of the members. And in all of these associations, this is an enormous amount of biodiversity, but there are a few trends that you can identify. And one of these is that symbiosis dramatically shapes host and symbiote biology. And it does this in a few definite ways that I'll detail quickly here. One is that it helps evolve novel lifestyles and niches. So here I've shown a couple examples. For parasites, we see time and time again that parasites evolve really complex multi-host life cycles, and this enables them to distinguish different stages of growth and reproduction and enable them to reach higher population sizes among different hosts. In mutualistic associations, you literally see the formation of novel niches, such as has occurred at the hydrothermal vents. So at these sites, superheated seawater that's been pushed through the seafloor due to the tremendous pressure at those depths comes back up and it contains reduced chemicals that then bacteria are able to oxidize and use the electrons to fix carbon dioxide and make ATP. And through association with these bacteria, animals can now colonize these niches. So this is a brand new niche for these host species, but it's also a brand new niche for the ecosystem as well, because now there's enough biomass to support heterotrophic communities that can feed on them. We also see time and time again loss of functions due to symbiosis. So these may be functions that are no longer needed or the host provides. An example for parasites or mycoplasma, intracellular bacteria that have lost their cell wall and now just have a membrane separating them from the environment. And we see in mutualistic associations time and time again that there's a complementation that arises between the biosynthetic pathways for compounds that are needed and are provided by the symbiosis. So here's an example involving aphids and their intracellular Buchnera bacterial symbionts. The aphids now live on a diet of sap. They don't obtain the amino acids that they used to obtain from their diet source, and so now they need these bacteria to help them complete the amino acid synthesis pathways. And lastly, we see often that developmental programs are altered in these associations. So in more pathogenic bacteria, such as reproductive manipulators like Wolbachia, Spiroplasma, certain Rickettsia lineages, that they're transmitted through the female germline, and they can cause sex ratio distortions because that will enable them to reach higher frequencies in the population, being a maternally transmitted bacterium. In symbiotic or mutualistic associations, such as occur between the bobtail squid and its bioluminescent Vibrio fischeri symbionts that it houses in this ventral organ. It uses these bacteria to produce light that counter-illuminates it, so predators from below can't see it. It's not a shadow against the moonlight from above. And amazingly, these bacteria are acquired from the environment every generation, and they actually induce the formation of the light organ that houses them. So they have to be recognized, and through signaling cascades actually induce the morphological development of this organ. So as these examples show you, one of the common trends among them is that host reproduction and symbiont reproduction often are linked and manipulated because the symbionts need to be transmitted between host generations and acquire the association reliably over time. And there's a diverse way among symbionts that this is accomplished, but basically you can boil these down to 3 different situations that exist along a gradient. At one end of the spectrum, there's strict horizontal transmission. In these situations, the host is born without bacteria, and it acquires those bacteria from a free-living population of bacteria or bacteria that travel through the environment from contemporary hosts. And one example of this is the tube-worm, Riftia pachyptala, that occurs at hydrothermal vents. It's a free-swimming, free-feeding larva. Upon settlement, it loses its mouth and gut and then acquires its symbionts through its outer body wall from the seawater. At the end of the transmission node spectrum, you have strict vertical transmission, and in these situations, the host reproduces and contains its symbionts within it throughout reproduction and development. You never find the symbionts free of its host, and they have to be coordinated with these host processes. And a great example of this is in the pea aphid. Its Buchnera bacterial symbionts are transmitted from adult symbiont housing cells to the embryo during parthenogenesis. And this also occurs with sexual reproduction in that phase of aphids. And then in between these two extremes, there are mixed modes of transmission, which incorporate some amount of both modes. And this can be associations that are vertically transmitted all the time, but occasionally there's horizontal transmission events that bring new symbionts in. Or it can be coming from the other end of the spectrum, where you have a horizontally transmitted bacterium that's beginning to evolve vertical transmission, but it's a low efficiency, and so it really needs horizontal transmission to ensure every host gets its symbionts. These modes of transmission are really important for understanding how these associations are perpetuated over time, but they're also important for understanding how the cost-benefit trade-offs evolve and how pathogenic or mutualistic associations arise. And you can think of this basically just in terms of fitness overlaps. Under strict vertical transmission, host reproduction is absolutely required for symbiont reproduction. And so their fitnesses are linked. One cannot do well without the other. And in contrast to that, under strict horizontal transmission, the host only needs to survive long enough for the bacteria to reproduce and make it out to colonize to the next host. You often see horizontal transmission in pathogenic associations and parasitisms. And this prediction is borne out with the data. So this is a recent meta-analysis of the literature looking at when you cure a host of its symbionts, how often do you see dramatic reductions in host fitness due to the loss of symbionts, and how is that partitioned by transmission mode? And so as you see here, vertically transmitted associations experience much larger fitness drops when you remove their symbionts than horizontally transmitted ones do, suggesting that this is one of the things that drives the cost-benefit trade-off. In addition to this, though, there's many other things that transmission mode affects and why I'm really interested in this aspect of symbiosis biology. One is that symbiont transmission modes necessitate molecular and cellular adaptations to enable the mechanism to work. So under horizontal transmission, bacteria, the symbionts, they need to be recognized uniquely from all of the other bacteria in the environment so that you don't accidentally take up a pathogen. And as I've described previously, this happens through very selective signaling cascades between the bobtail squid and its Vibrio symbionts, but similar interactions occur between legume plants and rhizobia bacteria that then are taken into the root and form a nodule where the bacteria can fix nitrogen. And then under strict vertical transmission, you have similar types of cellular recognition processes occur. So this is another example from the P aphid. To transfer to those embryonic cells from the adult host cells, there's actually an extracellular phase. So the symbionts need to be recognized on the extracellular level as well, but then they also need to be integrated during host development. So you can imagine there's a lot of mechanisms that need to evolve pretty tightly for this to work properly. And lastly, I'll mention, I'm going to go into this in much more detail later, but symbiont transmission mode dramatically influences the evolution of symbiont genomes. And this theory has been established since the late 90s when we first started sequencing bacterial genomes. But you basically see this trend that with increasing degree of host association and duration of the association, you see a degradation in symbiont genome size, getting towards the organelle end of the spectrum, like mitochondria and chloroplasts that have lost a lot of their genes and only contain a very minimal set. And so, as I hope all of these examples have illustrated to you, transmission mode affects many, many different processes that are really at the core of symbiosis biology. And these different impacts are felt across the entire time scale of the symbiotic association, from initial recognition mechanisms happening at the cellular level to speciation events and long-term processes happening over millions of years. But despite how important transmission mode is to understanding symbiotic interactions, most of what we know is from a select few associations that have been able to be worked up in the lab that are typically at the opposite ends of these spectra. So we know a lot about horizontally transmitted pathogens and these horizontally transmitted mutualisms I've talked about previously. We know a lot about vertical transmission in the aphid, but the interactions between hosts and symbionts in associations that are intermediate between these, that are mixed mode transmitted, or maybe sometimes are mutualists, sometimes are pathogens, we know less about these. And I think that studying these types of associations really offers tremendous insight into how these associations evolve and function. And this is for two reasons. One, mixed modes offers this opportunity to be able to disentangle the mechanisms that enable integration with host development for vertical transmission and also novel infection through the environment, the horizontal transmission component. And not only are mixed modes important just as a model, but in a meta-analysis I did recently, looking at literature going back to the early 1900s, I found that in fact mixed modes are pretty abundant in nature. They are about a third of interactions. So understanding these means understanding a missing component of symbiosis biology. So today I'm going to tell you two stories to get at this effort of understanding how mixed modes impact the biology and genome evolution of hosts and symbionts. First, I'll tell you a story about how symbionts localize to the germline using Drosophila melanogaster and Wolbachia, an alpha proteobacterium that is transmitted through the germline. And then I will look at the genomic consequences of mixed transmission mode over long periods of evolutionary time using a bivalve system. So first, starting off with the Drosophila Wolbachia Association. This is work I've done in collaboration with my postdoc lab, the Sullivan Lab at UC Santa Cruz. This is a really ideal system because, so Wolbachia is really fascinating on its own, but its host, Drosophila melanogaster, has all sorts of amazing tools and systems. We have in vitro, in vivo systems, genetic knockouts. It's been a dream working in a model coming from a non-model originally. So starting here, Wolbachia is ideal for looking at mixed transmission modes because it does exhibit different degrees of horizontal and vertical transmission depending upon the evolutionary timescales you're looking at. So here on the left, this is a tree for the mitochondria and Wolbachia from Drosophila melanogaster populations. And on these timescales, these pretty much evolve together. They're always transmitted through the germline. You have pretty strict maternal transmission as the congruence between these trees shows. But on longer time scales, as is shown here for this butterfly phylogeny, the Wolbachia doesn't map strictly onto the butterfly phylogeny. And in fact, there have been a lot of host switching events that have happened. And when you look at the genome size for Wolbachia, they're about half the size, a little less than that for free-living bacteria. So they're definitely intermediate along the spectrum of transmission modes and genome degradation. And they're enormously abundant in nature. So somewhere between 30 and 60% of arthropods are infected with Wolbachia, depending on how you do the math. And all filarial nematodes are infected. They're, in fact, obligate for these worms. If you remove them, the worms die. Some of these cases are obligate, many are more facultative, but understanding them means understanding probably one of the largest epidemics in nature. And so getting back at this question of how symbionts coordinate with the host development in order to reach the germline and make it onto the next generation. This is a pretty complex process. As I've shown in this cartoon, during host development, there's many cell divisions that the symbionts might not make it into the proper lineage of cells to then make it to the germline directly and instead go to the soma, or maybe they need to go to the soma to confer the function. And so how do they make it back? How do they coordinate with these processes is a pretty complicated question. And in fact, the going to the soma and then having to make it back to the germline, although it's not the most parsimonious series of steps, it does seem to be the most abundant in nature. So I did a review recently looking through the literature at all of the different associations we've done cell biology on to figure out how the symbionts are getting to the germline and adult hosts and where they go during development. And the vast majority of them first go to a somatic cell lineage and then have to migrate back to the germline or to the offspring at some point later in development. Very rarely do you see them segregating to the germline early on and maintaining two populations throughout development. And in Wolbachia, this also seems to be the case. So work from my postdoc lab previously has shown that in filarial nematodes, during cell divisions in early development, the Wolbachia actually segregate to a somatic lineage and then have to migrate back to the germline before reproduction. Something similar does appear to be going on in Drosophila melanogaster. We don't have all of the developmental time points yet, but they localize to the pole plasm, which will form the future germline in the next generation, but you can also find them throughout the soma in development. So there's probably a mechanism for them to, in case they don't segregate with the oocyte as it's dividing from the germline stem cell and developing, they could make it back in from somatic tissues at a later point. So to test this idea a little more directly, we performed experiments in cell culture. So we have Drosophila melanogaster cells that have been immortalized. We can just keep them in flasks, split them every week. They are naturally infected with Wolbachia. So the fly that these were derived from contained Wolbachia. And so this experiment just looked to see if cell-to-cell transfer is possible and on what time scales does this happen over. And so we exposed uninfected cells to infected cells through a transwell membrane that only bacteria can pass through, and then looked at the infection frequency of those uninfected cells over time. And sure enough, within hours, we see transfer of Wolbachia through the medium to uninfected cells. And these actually can form stable infections. So I have one now that it's been going since 2017. So this is a real process that can actually result in stable infections. Looking at mechanistically what might be underpinning this, it looks like they're using a mechanism similar to what pathogenic bacteria use to get into new host cells, where they plug into clathrin-coated pits. So when we knock down clathrin with a chemical that inhibits this protein, you see that there's a reduction in the amount of Wolbachia that's transmitted to those cells. And with these electron micrographs, we've shown that plugging into this pit seems to induce the formation of actin filaments that make pseudopodia that come up and wrap around the cell and take it in to the host cell. And this is very similar to a zipper mechanism used by many pathogens. So I've got a lot more work going on this right now. I'm making new cell lines from different species, different strains, but just preliminarily, it looks like these cell-to-cell transfer mechanisms might help mediate the horizontal transmission components. If they have to do these processes to make it to the germline normally, and they do them so readily between somatic cells, then perhaps this could underlie the horizontal transmission capabilities of these microbes. But to get at this other question, a little bit more finely, how do they integrate with host development without disrupting it? I used the Drosophila oocyte as a model system to test this question. And this is because, as I briefly mentioned before, Wolbachia exhibits really unique and distinct localization patterns throughout host oogenesis. So here I've shown, this is an ovariole from a Drosophila ovary. There's many ovarials in an ovary. And up here is the germline stem cell, which divides and then goes through transit amplifications to produce these nurse cells, 15 nurse cells plus one oocyte. And the nurse cells support the oocyte during development. And during these stages, Wolbachia is present and exhibits these unique patterns. So in early oogenesis, they have this anterior localization pattern. And then moving to late oogenesis, they have a posterior localization pattern. And this is coincident with the germplasm, which will form the future germline in the embryo. This region gets enclosed in pole cells that then migrate in to the germline. And we know from prior work that actually it's host motor proteins that mediate this transport. Motor proteins don't just transport host cargoes, but also can transport bacteria. And we know this this was some work about 15 years ago now, from my postdoc lab, finding that dynein heavy chain confers this anterior localization. So here, if you depolymerize the microtubules, or if you reorient the microtubules, or if you knock down dynein heavy chain or the linker protein dynactin, you see that the anterior localization is dissipated. Similarly, the posterior localization is conferred by kinesin heavy chain, and this is due to the microtubules changing polarization during development, but at this stage, plus N transport lands them at the posterior pole. And so similarly, if you get rid of the microtubules or you knock down kinesin heavy chain, you abolish this posterior localization pattern. However, when I joined the lab, we didn't know exactly how Wolbachia was attaching to this motor protein. Usually the host uses a linker protein, usually kinesin light chain, forming the canonical kinesin 1 unit to transport host cargoes. But we weren't sure, nobody had done the experiments to see if that's what Wolbachia was using. So that's where I started. So a priori thinking we knocked down kinesin light chain, it's probably using the whole unit, so we should see less Wolbachia at the posterior pole. But actually, that's not what we saw at all. We knocked down kinesin light chain and found that there's actually an excess of Wolbachia transported to the posterior region and the posterior pole relative to wild type. And so this was really striking. I think you all can see it, but Wolbachia are these little bright points, and I've shown you here an uninfected and infected just to help orient you with these images. But this is a really striking phenotype. This is, usually we don't see changes in the amount of Wolbachia that are transported when we knock down genes to this degree. And what it implied to us was that perhaps kinesin heavy chain is limiting for Wolbachia, and that by getting rid of kinesin light chain, we freed up the use of kinesin heavy chain. But there could also be regulatory or signaling dynamics that have changed due to the lack of kinesin light chain. So to test this more directly, I overexpressed kinesin heavy chain directly, and found that indeed this phenotype is recapitulated when we overexpress kinesin heavy chain with different promoters and different copies of extra kinesin heavy chain in the genome. And this is even more significant relative to wild type than the kinesin light chain knockdown. And it's just interesting to note, we can also recapitulate this phenotype if we free up more kinesin heavy chain just by eliminating one of its functions. So kinesin heavy chain can also be used to produce microtubule sliding, which causes the cytoplasm to turn in the oocyte and redistributes all the different components in the cytoplasm. And so if you eliminate that function by deleting those residues, you actually get more Wolbachia transported to the posterior pole. And so that led me to propose this model for Wolbachia transport, where Wolbachia is competing with kinesin light chain for access to kinesin heavy chain, but it's a pretty poor competitor for these processes. And so if you increase the kinesin heavy chain concentration, you get a proportional increase in Wolbachia localization because more is available for it to use. And importantly, this is a unique trait for these bacteria. So if we overexpress kinesin heavy chain, which is shown up here, and look at the distribution of pole plasm components, which is in green here, you see that while there is some qualitative difference in the localization, there isn't more pole plasm transported relative to wild type. And that indicates that Wolbachia is unique in its limitation of access to KHC. And it has a unique dynamic with this motor protein relative to host cargoes. And that led me to propose that this is sort of the evolutionary dynamics that are underlying this association, where you have a bacterium enter a host for the first time. It's poorly evolved to using those host proteins. Even if it came from a related host, it hasn't been co-evolving with the proteins like the host has. But over time, it can develop a strong interaction for host proteins, but then that might feed back on the host and cause the host to not function quite as well, which then results in a loss of fecundity for the bacteria, because if host development's interrupted, then bacterial reproduction will be interrupted. And so it probably is a process where the affinity is limited by this interaction. But what linker does Wolbachia use? We're still left with this question. And so thinking about how dramatic of a competition it seems Wolbachia has with kinesin light chain, I thought that perhaps it's making a kinesin light chain-like protein. And so I looked through, by BLAST, the Drosophila strain of Wolbachia's genome, the WML genome, for proteins that share sequence homology, or it's probably mimicry in this case, with the Drosophila kinesin light chain protein. And sure enough, I found a protein that has strong sequence similarity at specific regions, and many of these are TPR repeats, which actually form the cargo binding region of the protein. And so there's sequence similarity, but also there's structural similarity. So I predicted the protein structure of the Drosophila KLC protein and the WML protein using the human KLC crystal structure as a model, and found that, yeah, these have very similar structures overlaid on each other. You can see they're almost identical. And since structure confers function, this is potentially what Wolbachia is using to link to kinesin heavy chain and be transported through the cell. Importantly, we do see this protein, orthologs of it in other Wolbachia strains. But they're very fast evolving, and so I wanted to see if there was a correlation with the presence of this protein and the ability to localize to the posterior pole. And for most of the cases that we have data for, there's a lot of missing data. People just haven't looked at this in these other organisms yet. But however, there is one case where there's a mismatch. The protein's present, but we don't see posterior localization. But when you look at the alignment here, you can see that the TPR repeats have been deleted, and it only has the N-terminal part of the protein. So this suggests what region of the protein might be needed. But further experiments are underway. I'm doing mass spec on pulled down heterologously expressed constructs. So this should be pretty exciting, but that's all I have for now. So in summary from this section, looks like cell-to-cell transfer occurs readily in these bacteria in their host cells in vitro, which implies it might be needed for normal vertical transmission and could potentially enable horizontal transmission between hosts. Wolbachia has evolved to not outcompete host processes during its transmission in localizing to the particular parts of the oocyte that will become the future germline. And the bacteria probably make its own proteins to confer some of these movements. So this is great. I think I've filled in a good number of the gaps in what we know on the cellular organismal levels about mixed transmission mode. But what happens over long periods of time, specifically to the genome evolution of these symbionts under mixed transmission modes. So leads me into my second section here. For this work, I used some chemosynthetic bivalves. I'll describe exactly what that means here in a second. But they basically are bivalves. This is a gill filament, a set of gill filaments from a bivalve that contain intracellular bacteria, which are colored here in red. And these make really excellent models to investigate this association because they're mutualistic, and you can find a variety of transmission modes among them. And so I did this work during my PhD with Colleen Cavanaugh at Harvard University, and I've continued it in collaboration with Russ Corbett-Deddick at UC Santa Cruz. Okay, so as I mentioned earlier, in symbiont genome evolution, over long periods of time, you see this progression towards genome degradation with increasing host association, vertical transmission, and mutualistic types of interactions. And this is thought to be the case for a couple of reasons. Mainly because symbiont transmission mode and host restriction and obligacy really restrict the gene flow of symbionts between host individuals. So if you imagine a host population moving through time here, and then the symbiont lineages, two of them just for an example, mapped on top, you can see under strict horizontal transmission that the hosts aren't really much of a barrier to symbiont gene flow among hosts. They might lock a symbiont in for a generation, but by then the next generation comes around, they could be in a completely different host, encounter different symbionts. These form pretty large populations. And then in contrast to that, under strict vertical transmission, the symbionts map tightly to the host lineage, and they're locked within the host lineage indefinitely. So, once these split, these two lineages will never come back into contact. And so that leads to very, very small population sizes. And in addition, every single time there's a transmission event, there's a transmission bottleneck. And so drift dominates in these systems because, your chance of sampling the full diversity is pretty minimal when you're bottlenecking down to a few hundred symbionts. And then under mixed modes, you see an intermediate state between these two, where the symbiont population sizes are intermediate size, and symbionts can occasionally come into contact with new host lineages and new symbiont lineages. And this is important because it determines the efficacy of selection. So under strict horizontal transmission, a deleterious mutation arises, and it can pretty quickly be purged from the population because there's another fully functional variant that exists that can come in and replace it in the other generations. Whereas under vertical transmission, that deleterious mutation arises, has a pretty good likelihood of going to fixation during that transmission bottleneck, and then all the descendants have that mutation and it can't be rescued. Whereas under mixed modes of transmission, you might have that deleterious mutation locked in, but it can potentially be rescued by a horizontal transmission event at some point. And so these are important processes to think about, because while that theory of endosymbiont genome evolution is primarily true for many, many associations, there's a good number in particularly marine environments that don't seem to obey these predictions. So despite many of these associations being really old and very obligately associated, mutualistic, in the marine environment you really don't see genome sizes much lower than 1 megabase, or about 30% the size of free-living bacteria. And on land, you see way, way larger extents of genome degradation, where the symbiont genome size can get down to about 4% of free-living. And in thinking about what could be underlying this, one of the things that occurred to me was that, these are drastically different environments that these symbioses occur in. And, perhaps the seawater provides a nice medium for bacteria to be transmitted through. And so in that analysis, that meta-analysis of all the different symbiont transmission modes, I looked for an association between habitat type and transmission mode, and found that indeed there's a deficit of vertical transmission in aquatic habitats, whereas there's an excess of vertical transmission in terrestrial habitats relative to chance. And so this suggests that perhaps it's ongoing gene flow via horizontal transmission that's mediated by the seawater that's preventing these genomes from degrading completely. But to test this question, I needed a model system, and so I used chemosynthetic bivalves from 3 different families. And I'll tell you why in a minute, but just to kind of introduce you to what these organisms are. These are mussels that occur at hydrothermal vents and cold seeps. These are giant clams that occur at hydrothermal vents, and these are little protobranch bivalves as a basal group of bivalves that occurs in coastal sediments. They all have intracellular bacteria that are able to oxidize inorganic compounds such as sulfide. These also have a methane oxidizing symbiont, and they use those electrons to fix carbon dioxide, make ATP, basically feeding the host. Most of the nutrition comes from these processes. And these groups are ideal for testing these questions because they have distinct yet conserved methods of transmission. So the mussels are strictly horizontally transmitted. They acquire their symbionts brand new every generation. The solomyids have mixed transmission modes. As you can see here, they're contained in the ovary. They're found in larvae and even in the developing gill buds of larvae. And then the vesicomyids are mostly vertical. So I say mostly because on long periods of evolutionary time, you can see there have been a couple of host switching events, but for all practical purposes, short periods of time, they're just transmitted through the germline, and you can see them here in the oocyte. And when you look at how they're related to one another, the effect that these transmission modes has on their evolution is pretty clear. So the vertically transmitted vesicomyids are all a monophyletic group up here. The mussel symbionts are interspersed along the tree with free-living bacteria falling out between them, and then the same thing for the mixed transmitted solomyids. So to test these questions, I had to make some data and collect some samples. So I collected some of the solomyid velum samples myself from the east coast of North America, did some freezer diving for the other solomyid species and the Calyptogena species. Fortunately, a lot of samples have been collected from the 90s on up to the mid-2000s. And then a collaborator, Roxanne Beinart, helped with the mussel samples. And so using these, we sequenced the gill DNA, which contains nuclear mitochondrial symbiont genomes, and performed de novo assemblies using nanopore and Illumina data. And then we also made population data sets with Illumina sequencing from several individuals from each species. So first, I'll show you what these assemblies look like. So as we would expect, the vertically transmitted symbionts have smaller genomes than the mixed and horizontally transmitted symbionts do. These are pretty close to free-living. Free-living sulfur-oxidizing bacteria have genomes about 3 megabases in size. These are, a half to a third that size. Great, but what is keeping this from eroding further? They all hover around this size. There's probably some ongoing gene flow. So we're next to look at the population data. First, I'll show you a method of looking for evidence of horizontal transmission. So under strict vertical transmission, symbionts and mitochondria should be co-inherited through the oocyte, so they track the maternal lineage, and the genealogies of mitochondria and symbionts should be completely concordant due to that shared cytoplasmic history. But I'd like to point out that this test is very easily rejected due to like a little bit of horizontal transmission. So theoretical work has shown that even very, very low rates of horizontal transmission can erode this signal. So this is a first level look at whether there's horizontal transmission ongoing in these species. And indeed, that's what we see. So here I have shown you genealogies of mitochondria on the left, symbionts on the right, with white lines connecting the mitochondria and symbionts from the same host individual. And you can see that these lines cross pretty frequently, indicating that there have been switching events. The topologies don't map onto one another. And there have been as many events here as you see in the mixed transmitted and horizontally transmitted symbionts. Okay, great. So clearly, genome erosion can occur with ongoing horizontal transmission. Maybe that's what's limiting the erosion of genome size. But to look at this in a little more detail, next I looked at the structure of these populations to see how much of the variation is shared between host individuals and how much of it's private to a host individual. Because you would expect that under low horizontal transmission rates, each host is basically an island, and not much of the variation is shared between hosts. In comparison to high horizontal transmission rates, you should see, basically many of the variants contained within one population are represented in another population, and these are represented by high FST and low FST values, respectively. Looking at this for the symbionts, we see what we would expect. So FST values are very low, indicating a lot of mixture between host individuals for the horizontally transmitted and mixed mode transmitted symbionts, whereas FST values are very high for the vertically transmitted vesicomyades. But there could be variation that we're missing here. If there is variation within a host that we didn't happen to sample in another host that we picked up from the locality, then we wouldn't be picking up on it here. So next I wanted to look at what the patterns of diversity look like within host tissues. And to do this, I plotted the allele frequency spectra for the symbiont within host populations. So here are just 3 representative individuals from 3 of the 6 species, but they all looked consistent for their transmission mode types. So I'll just show you the examples here. So starting with the horizontally transmitted mussels, this allele frequency spectrum with an abundance of low frequency alleles that taper off towards higher frequencies, this is pretty much what we expect for free living population at mutation selection equilibrium. So this is great. This is clear that these guys are forming large diverse populations that are mixing between hosts and recapitulating a lot of that diversity on the within host level. Then coming over here to the vertically transmitted association, if you look at the y-axis, there's like no variation in here. These are pretty much clonal. The few SNPs that I detected could even be alignment errors potentially, because those are difficult to control for. So there's not much going on in these vertically transmitted symbionts at all. The populations are homogeneous. And then in contrast, the mixed modes have really crazy stuff going on. These have all sorts of different patterns of diversity with intermediate multimodal spectra. I'm not going to get into it today because I don't have time, but I have a paper about this, and it basically looks like there are mixed transmission events resulting in mixed populations in these host tissues. A recombination event happens. This isn't enough variants to comprise a whole new haplotype. Recombination event happens between the resident genome and the new genome, and then that variation is inherited, and that's how you get this pattern in host tissues over time. So this is striking. This indicates that horizontal transmission may occur, but it's very, very rare. However, it's important to think about recombination, because even if horizontal transmission is rare, if it occasionally occurs, then that new haplotype could recombine with the existing haplotype and potentially rescue any deleterious mutations that have come up since the last horizontal transmission event. So to look at the rate of recombination, we performed the 4-gamete test to fit values of recombination to a coalescent model. And basically the logic here is that due to the improbability of the same mutation happening twice and the linear nature of the chromosomes, you shouldn't see all 4 pairwise possible combinations of gametes unless recombinations happened. And sure enough, we see a difference in recombination rates, but the vertically transmitted vesicamides aren't at 0. So they've lost the predominant recombination gene REC-A, but it still seems like they're capable of non-REC-A mediated methods of recombination, because we do detect a fair number of sites that fail the 4-gamete test. But in comparison, the horizontally transmitted and mixed mode transmitted symbionts have really high recombination rates. And just to kind of show you how high of recombination rates these are, here's a little more data from one of these species, Solomyavelum, a mixed transmission mode organism. And this is just plotting the linkage between alleles against distance between them in the genome. So if recombination is ongoing and frequent, you should see a decay in linkage with genomic distance because of the increased probability that recombinations happened as distance increases. And indeed, that's what we see here. This is highly significant, and all linkage is eroded by about 80,000 base pairs. So these processes clearly are ongoing, very dramatic, and for the vesicamides, potentially can help rescue their genomes and keep them at that slightly larger size. However, there's other things that recombination can shape in the population, and one of these is the efficacy of selection. And so under a lack of recombination, you basically get clonal interference that results because haplotypes are locked together. You can't select a beneficial mutation independent of the rest of the genome, and you can't purge a deleterious mutation independent of the rest of the genome. And so you end up selecting for entire lineages that win out compared to the others. And that results in an excess of rare alleles when you plot the between host allele frequency spectrum, and long branches with few mutations shared between the terminal lineages. And this is in contrast to a freely recombining population. Looking at the symbionts, we see indeed it looks like clonal interference is happening in these vertically transmitted symbionts. They have an excess of rare alleles and really, really long terminal branches. We see a little bit of this in the mixed mode symbionts, which is kind of surprising considering how rampant their recombination rates are. But so this indicates that while recombination is happening, it's not happening very efficiently in these guys, enabling the degradation process to proceed, but only to a point. Thinking about how genome structure is impacted by these processes, in bacteria, recombination can induce structural changes such as inversions, deletions, and insertions. And so if you lack recombination or recombination is depressed, then you should see less of these structural changes. And indeed, that's what we see for the vertically transmitted vesicamides. So these genomes are largely synthetic. You can count the number of inversions and rearrangements that have occurred, and this is in comparison to these other two groups that they're so dynamic. There's even horizontal gene transfer ongoing in this species here. Those genomes are moving pretty fast and pretty dynamic. And in all, this suggests that these processes of horizontal transmission and recombination are able to maintain genomes in this intermediate state of degradation, that as long as you have enough of these processes going on, you can have some degradation occur, but it doesn't go all the way down to the small sizes we see in the terrestrial endosymbionts, as long as it's maintained. And so in summary, genetic signatures of vertical transmission are eroded pretty quickly. Horizontal transmission signatures predominate. Host tissues can harbor substantial amounts of symbiont diversity, or they can be clonal, depending on the rate of horizontal transmission. And the rate of symbiont genome decay is determined jointly by the rates of horizontal transmission and recombination, and may underlie the lack of degradation of symbiont genomes in the marine environment. So hopefully I've covered quite a bit of the spectrum for you guys today. We understand a lot more now about the cellular mechanisms occurring on the host level for symbionts to be able to get to the germline and move between somatic cells and germline cells. And then on long periods of time, vertical transmission does cause genome degradation, but horizontal transmission is able to prevent that process and sustaining intermediate levels of degradation. So with that, I'd like to thank everybody I've worked with, all my collaborators. Thank you for your attention, and welcome any questions.
Questions?
Yeah.
Hi.
Hi.
I love your talk.
Thank you.
I was wondering about your hypothesis that you have an accumulation of deleterious variants in the vertically transmitted organisms because of lower ME.
Yeah.
So I think it's a great model. But then I'm wondering, if you have an accumulation of deleterious variants, then wouldn't that also make it susceptible for replacement?
Yeah, and that does happen. Yeah, so there are many sap-feeding insects, and like Hemiptera, there's a good number of examples of this where the symbiont genome has become eroded to the point that it can't fulfill the primary function that it was providing for the host, and so a second symbiont has to be taken up, or the symbiont has to be replaced. There's two ways of solving that problem.
So, but then it no longer becomes a exclusively vertically transmitted organism.
Well, the new ones tend to go to vertical transmission too. They just take up a new one and integrate it. The short time scale dynamics of these processes aren't really well understood. Like, we're looking at these things millions of years after they happened and inferring replacement based upon what all the other lineages have. But yeah. Yeah.
So earlier on, you had a discussion where the host genome was changing in response to the attack by that parasite bacteria.
I haven't been able to look at the host side of things yet with this, unfortunately.
Well, I was going to ask, because you were talking about some of your cell lines and your cell affected, and presumably they have one genotype, and you have cell lines in your cell which have never been affected. Because I assume they've never been affected.
Right. Oh, so like S2 cells versus like infected cells that we've made.
Ancestry sometimes long ago, something was infected.
Yeah.
Lost it, I don't know. Comparison of their kinesin light chain genomes and other parts. Does that show a difference, or are they still the same?
I mean, I suspect it's the same. So these proteins are really conserved between Drosophila species. Like in comparison to, there's reproductive genes that are moving pretty fast between like simulins and melanogaster, but the motor proteins are pretty highly conserved. So there could be weird things going on with the cell lines. Like, I know the S2 cells have become, they're polyploid to some degree. Like, there's weird things that happen in the immortalization process, but I wouldn't expect it, but it might be worth looking into. Just, it's cheap enough to sequence them. Yeah, good question.
I have a question. So in the first part of the talk, when you mentioned, so you have Drosophila and Wolbachia that they have these proteins that are very similar and presumably serving very similar purposes.
Yeah.
Are these coming from like homologous genes moving in parallel, or genes of different origin and converging?
I think it's convergence. I think it's mimicry, because you do see this in intracellular pathogens where they mimic host proteins, and the the sequence similarity is only regional, and it's on the amino acid level. So yeah, so I'm suspecting they've kind of stumbled into this, this evolution of it's not through shared descent or horizontal gene transfer or something. Yeah, that's my guess at any rate. Yeah.
I thought your work on the mimicry of the host proteins was very elegant.
Oh, thanks.
And my question to that is this something that's going to work, mostly with invertebrates, or what are the implications when you do have more self-non-self discrimination in the host itself?
That's a good question, because you don't see, like, there's intracellular parasites in vertebrates, but you don't see intracellular mutualists. And kind of the hand-wavy argument I've heard over the years is just adaptive immunity doesn't permit it to happen. So I don't know if I can answer that question, but I do know that mimicry does happen between human pathogens and their host on the intracellular level. So yeah, I mean, at least for those sorts of interactions, yes. Yeah. Yeah.
Awesome research.
thanks.
I just wanted to know if the symbiont's function in the host dictates its mode of transmission.
So, not that I can tell, because so symbiont functions are, like there's general categories you can bend them into, like nutrition, defense. You see horizontal transmission occurring in all of these, and you see vertical transmission occurring in all of these. Now, I think there is a correlation with the benefit, whether it's a costly interaction or a mutualistic interaction, but I don't think the particular function is predictive of the transmission mode. Yeah. Yeah. Yeah.
So there's one very specific mode of mixed mode transmission that I was curious about the signatures of, where the endosymbionts are completely inherited by only one of the progeny. And like if there's fusion, one gets the symbionts, and the other one has to acquire them from outside.
Yeah.
So it's like you kind of have a lineage that's going through, that's fixed. But on the other hand, there's also,
Right.
like it's a boundary thing. So what are the signatures that you see?
Oh, well, so we do see some of that in wild Drosophila populations. So in the lab, maternal transmission is 100%. But if you go out into the field and do these assays, it's not. It's really variable. And in fact, so Brandon Cooper, he's a PI at University of Montana. He's gone out and done these assays on a bunch of different species across like islands, and it seems to be habitat dependent, strain dependent, host species dependent, where you can get 50% maternal transmission or higher rates. So we're not clear on what is actually regulating that, but we do know that host diet affects symbiont titer. So Wolbachia is at a higher density when it has less yeast in its diet than more yeast. It's through like an insulin signaling cascade, and so perhaps that's part of how diet plays into it, because if there's fewer Wolbachia overall in that germline stem cell and in the other surrounding cells, then you could imagine there might be an oocyte produced that lacks them, just because there weren't enough and then they didn't make it in later. But yeah, these are all great questions. Yeah.
We might have time for one more question. Thank you again.
Yeah, thank you.